All right, so let's look at how a muscle fiber contracts. So the decision comes, it's a voluntary muscle, skeletal muscle condition. So we're talking about activation by the brain, goes down the spinal cord to the motor neuron, which activates the muscle fiber. The neurons in the muscle cells are excitable cells, so that's part of their form with their anatomical structure, right? They have the ability to be excitable, the excitation, right? So they can change the resting membrane potential voltages. So the resting membrane has a charge, and that's measured in volts, so that's the voltage. And a change in the regular voltage is what causes the action potential to move. So the action potential crosses the neuron to the muscle cell. From the neuron to the muscle cell with the neurotransmitter acetylcholine. That's the messenger, the chemical messenger. Ion channels. So recall that ions are charges. So we're looking at net positive, net negative charges. So that is the charge is what's going to change the membrane potential. There are two classes of ion channels that are used. The chemically gated ones are opened by such things like the acetylcholine example that I just gave you. And then the voltage gated ones are opened by a change in the voltage. So, okay, so it's the same thing. But here, let's look at it. So here's a chemically gated ion. Acetylcholine binds to the receptor, and at the receptor site, it says for it to open. In a voltage gated ion, again, it's a change in membrane potential that causes the gate to open and then allow the information to come into the cell, skeletal muscle. So here's our brain, right? This is where we decide to move, travels down the spinal cord to the neuron, and the neuron travels all the way down until it branches out here with axons. Or this is the axon, I'm sorry. And then each axon divides into branches. So the axon is just the thread-like projection. So if you have the cell body, the motor neuron cell body, the axon is the thread-like projection that then goes out to the muscle fiber. It branches at the end, and then here we have our synapse, and where the action is at the neuromuscular junction. So each muscle fiber gets its own axon terminal. And I just said that the neuromuscular junction. This is also called the motor end plate. Those are the same thing. You'll see NMJ for the abbreviation for neuromuscular junction, but we're talking about the interface between the neuron and the muscle. There it is, the neuron and the muscle, the interfacing at the junction. Okay, so this is great. I like this because it's straight definition, but the axon terminal, or the end of the axon, is separated by a gel-filled space called the synaptic cleft. So I just like that definition, gel-filled space, but that is what the synaptic cleft is. There's synaptic vesicles, which are like these little balls or containers that have the neurotransmitter acetylcholine in them. So those are stored within the axon terminal, which is like a bulbous, that kind of, it looked like cauliflower landing on the skeletal muscle fiber. So there's some junctional folds that have the acetylcholine receptors, and those are happening within the sarcolemma. Recall the sarcolemma is that wrapping around the muscle fiber that we looked at previously. Neuromuscular junction consists of axon terminal, synaptic cleft, and the junction folds. So when we talk about that, what is the neuromuscular junction by definition? It's the terminals, it's the cleft, the gel-like space in between, and then the junctional folds of the sarcolemma. That's where we're looking right now. So here it is, the neuromuscular junction as stated, right? The synaptic, the axon terminal. This is the synaptic cleft in here. It's space, right? And then here are our inner infolds, right? The last piece, junctional folds of the sarcolemma. So when we looked at the picture before where the muscle fiber was wrapping the cell, when we looked at it before, we couldn't quite see it in this detail that there was those infoldings. It just looked like a wrapping around the muscle. But now we're getting in real close to look at what's going on here. So next we'll see how these gates need to open to receive the neurotransmitter acetylcholine. So we're going to see what happens to make skeletal muscle contract. So there's certain events, there's a cascading of events at the NMJ. The muscle fiber becomes excited. There's this thing called excitation contraction coupling, and then the cross bridge cycling, which again is, recall the myosin head and the actin filaments sliding past each other and contracting. So that's the cross bridge cycling. Okay, so what's going to happen at the neuromuscular junction? AP, action potential, arrives. The voltage gated calcium channels open, so that again, it's like a battery. When you have the charges flip, it causes a change, and the channels open, so that's what allows the calcium to enter. Calcium causes release of acetylcholine, and then they go across the cleft, and that has to do with the charges within that space at that time. And acetylcholine diffuses across. It's going to cross the cleft. The sodium chemical gates are going to open once that neurotransmitter is received, and the sodium will enter, resulting in end plate potential. So there's a charge there, and then that charge is what's going to propagate the contraction. Acetylcholinesterase is the enzyme that degrades the acetylcholine to stop the event. So let's look at it again. I'm going to say the same thing, but with pictures in front of me while I do it. And okay, so here is the axon, right? And then the axon terminal at the fiber. So here's the here's the axon terminal. The nerve impulse reaches the neuromuscular junction. Okay, so it has arrived to this space. That's what it means. And then the acetylcholinesterase is going to be released. So when the action potential arrives, right, the calcium here is what's going to enter in to the axon terminal, right? That's going to affect this these little vesicles of acetylcholine. They're going to go through exocytosis. They start to move with the influx of calcium in. They start to move to this end, and they exocytose out, and that was what releases the neurotransmitter across the synaptic cleft. So this is just saying it that the action potential arrives. Here's the calcium channel opening, and then that's how the calcium is going to move from outside this area to inside this area. Just as I said, the calcium enters and the acetylcholine is released. It then crosses the neuromuscular junction. It's by diffusion at this point, so it exocytoses and then diffuses across the synaptic cleft. And then there's receptors on the sarcolemma here, which are those little purple things. Okay, so these are the receptors here, right, at the junctional folds, and they're closed. So when the acetylcholine then is received by the receptor and it attaches there, that's when those actually open. Okay, when they open, that's when we get an influx of sodium and potassium. So this is the sodium ions enter inside the membrane, okay, so into the cell, and the potassium ions exit, right? And this causes the membrane potential to change and then to propagate the action potential down downward, so it keeps the information flowing. We're going to look at this in more detail, exactly how that works. For now, we're just getting the cascade, the flow of how this works at the neuromuscular junction. So the last bit is just that the enzyme comes in to degrade the the acetylcholinesterase is the enzyme. It degrades the acetylcholine, right? And then once those are degraded here, they're no longer at the binding site, and that causes the gate to close again. So it stops more information or more of the influx, exflux from happening. All right, that's it for now.